Good morning. This is the promised review of malate dehydrogenase structure, allostery, and kinetics. My name is Ellis Bell. I'm going to share my screen so that you can see my slide deck. And we will proceed. So hopefully you can see my slides now. Actually, I'm not sure I've got it set right. Okay, so as I said, this is a review of malate dehydrogenase structure, allostery, and kinetics. I'm going to record the whole session, but I'm going to record it in 3 parts, just for convenience later on if you're trying to look for just the structure part or just the allostery part or just the kinetics part, so that you don't have to go through the whole thing if you don't want to. So this first part is going to focus on structure. And so on the top left-hand side of the screen is the typical dimer structure, which most malate dehydrogenases are dimeric structures. So here's the dimer structure. It's made up of 2 identical, chemically identical. I say chemically identical because each subunit has the same amino acid sequence, but does not necessarily have the same 3-dimensional structure and or dynamics associated with it. But they are chemically identical. Some malate dehydrogenases are tetramers, shown over here on the right. These, again, there are 4 chemically identical subunits, but they're not necessarily structurally identical from the 3-dimensional structure and dynamics. And I define structure as having various components, primarily the amino acid sequence, but the 3-dimensional structure and the dynamic aspects of that structure. And we'll talk about dynamics. So, talking about one subunit, each subunit has a Rossmann fold, also called Rossmann fold. It's actually discovered by Margaret Adams, and that's associated with cofactor binding. The Adams fold, if we start from the N-terminal, has a beta strand, loops round into an alpha helix, to another beta strand, and the beta strands are parallel to another alpha helix, to another beta strand, and then that connects to the 4th beta strand, and again it's a parallel beta pleated sheet, another helix, another strand, another helix, and then the final strand. Because there's symmetry in the cofactor, it's a dinucleotide, there's symmetry in the Adams fold. Okay, so if you put a plane of symmetry there, you can flip-flop either side and it superimposes on the other, and that reflects the symmetry of the cofactor. Okay, and this just shows at the bottom a typical cartoon-like structure of an Adams fold, a cofactor binding domain, a dinucleotide binding domain. Okay, one interesting thing about the cofactor binding domain is that on beta 2, i.e. here at the tip of beta 2, there is an aspartic acid residue that is common to all of the NAD-specific malate dehydrogenases. Some malate dehydrogenase can use NADPH. The phosphate on NADPH is at this position on the first ribose ring, the ribose ring attached to the adenine. And that is prevented by having this negatively charged aspartic acid here. So the NADP specific malate dehydrogenases have a little bit different structure around the actual cofactor site. The other thing that is common to all malate dehydrogenases is a so-called charge relay system. This is the active site. Here's the nicotinamide ring of the bound cofactor. Here is malate. And the malate is coordinated by several arginines that we'll talk about. But in terms of the business end of the malate, here is the OH of the malate. That proton is going to get abstracted by a histidine. This is the canonical histidine that is in the active site of all malate dehydrogenases. And then the hydride transfer occurs from this position to the nicotinamide ring. Now, the basicity of this histidine is impacted by an almost completely conserved aspartic acid residue that pulls and polarizes the proton on this side of the imidazole ring and increases the basicity of this nitrogen. Okay, this aspartate is completely conserved functionally. I think there's one malate dehydrogenase where it's a glutamate instead of an aspartate, but in all others, it's an aspartate. So that gives you just sort of a feel for what's going to happen with the chemistry and the cofactor binding. We're going to talk more about the active site region because there is a mobile loop that closes over the active site upon substrate binding that is critically involved in catalysis. So let's quickly just review some of the metabolic roles of malate dehydrogenase. Okay, it has over here on the right-hand side so-called housekeeping roles in cellular metabolism. It's part of the citric acid cycle, the TCA cycle, the Krebs cycle, as everybody except Krebs called it. And here's malate dehydrogenase. It catalyzes the reaction from malate to oxaloacetate with the concomitant release of NADH that is then used in oxidative phosphorylation by the electron transport chain. There's also a shunt that's known as the glyoxylate shunt across here that goes from isocitrate to malate. This is particularly important in plants. And as you'll see, there's a glyoxylate cycle in plants. This is the mitochondrion where the glyoxylate shunt occurs. Glyoxylate cycle is particularly important for fat metabolism in seed germination. And so malate dehydrogenase again plays a role in the glyoxylate cycle, catalyzing reaction from malate to oxaloacetate. In peroxisomes, there's often a peroxisomal form of malate dehydrogenase. So the form in the mitochondrial and the Krebs cycle is the mitochondrial malate dehydrogenase. In the glyoxylate cycle, it's often referred to as GMDH for glyoxylate malate dehydrogenase. And in peroxisomes, it's often referred to as PMDH. Again, it catalyzes the reaction from malate to oxaloacetate. And it's involved in the redox balance in the mitochondrion and the cytosol. In cytosol, there is a cytosolic form of malate dehydrogenase, and it's involved to help solve the NADH buildup problem in anaerobic metabolism, i.e. it converts oxaloacetate into malate and converts NADH to NAD. So it runs in the opposite direction to the direction in the other locations. Okay. In plants, there's a chloroplastic form as well that is NADP specific, but the chloroplast also has many of the other forms of MDH as well. But the often referred to as a chloroplastic malate dehydrogenase tends to be NADP specific. So bottom line is there are many different isoenzymes of malate dehydrogenase. So this is the amino acid sequence of malate dehydrogenases has been looked at extensively across the tree of life. This is a so-called sunburst representation of the relationships of various malate dehydrogenases across the tree of life. When you look at what people more usually look at, which is a clustal alignment and a guide tree from the clustal, you see there are different categories that I've referred to. So that's the NAD specific, there are NADP specific, there are cytosolic NAD specific. This is another class of NAD specific. And then there's the organelle NAD specific. And listed by the arrows are the various forms of malate dehydrogenase that the Bell Lab works with. And we've got clones of all of these, and in many cases, mutants of all of these. And we're happy to share if you're interested in developing either a research project or a cure based upon any of these. So these range, and I'm going to talk about a few of them as we go. This is P. gingivalis, it's part of the oral microbiome. There's a Candida albicans one down here somewhere that is also part of the oral microbiome, and you can see they're quite separated in the tree of life. There's the various Arabidopsis ones that are, this is the chloroplastic or NADP specific, but this is an organelle NAD specific. And there's sort of all sorts of everything in between. So there's quite a range of different malate dehydrogenases from different components of the tree of life available. And on the MCC website, there are many more than are listed by arrows here. These are just the ones that my lab, my cures work with. So when you do an overall clustal alignment, you find that there are 10 residues that are completely conserved across all malate dehydrogenases. And this just shows the location of them. Okay. This is the catalytic dyad histidine. This is the aspartate in the dyad. This is one of the three arginines. This is another one of the three arginines. This is the third of the three arginines. There are a couple of glycines that are involved. There's a proline, and there are two asparagines that are involved. This one is the base of the so-called mobile loop that we'll talk about. This one is totally separate from that, but those are the completely conserved residues. Some of them like the arginines, we've got a reasonable handle on what they do in the overall structure and function. The catalytic dyad, of course, we know what it does in the overall function, but the other ones, we're not too sure about what their complete role is. Okay, so I talked a little bit here about the catalytic dyad. Oftentimes people think, oh, it would be fun to mutate the catalytic dyad. Don't bother. We've already done that for both the histidine and the D193, the carboxyl group that's been published. And the crystal structure of the D193 N mutant has been determined in my lab by Wazo Myint in conjunction with Jessica. And the crystal structure shows that when you mutate D193 to asparagine, it does not change the overall structure of the protein. So oftentimes when you mutate something, it changes the overall structure, which makes interpretation of any impact difficult. But as shown here, we did the crystal structure and showed that the structure was not changed. More importantly, what was changed was the catalytic activity. There was a 660-fold reduction in the catalytic activity when you made the D193N. There's an almost 10,000-fold reduction in the activity when you take out the histidine and convert it to a glutamine. We think this is because when you take out that histidine and put a glutamine in there, imidazole, which is often used in the purification of the histag proteins, can rescue a minimal amount of activity. So it's not that the glutamine is catalytically active. We think this is due to rescue by imidazole in the buffer system. So I've talked a lot about the mobile loop a little bit, and in the 1-SMK PDB structure, this is our glycosomal malate dehydrogenase structure. It has a number of subunits in the asymmetric unit, even though it's a canonical dimer. The asymmetric unit, which reflects some asymmetry in the dimer structures, shows a flexible loop as judged by what's called the temperature factor from the crystal structure. And you have an elevated temperature structure through this region, and all except the black are empty subunits in that crystal structure. The black is what you see when you let citrate bind. And interestingly, citrate binds only to one subunit. And so this is the one subunit in the asymmetric unit that's got citrate bound to it. And you can see that the temperature factor is lowered down to a fairly low number, whereas this region have much higher temperature factors. Higher temperature factors are associated with local flexibility, and that's why this region is referred to as the mobile loop region. Okay, so this just summarizes the chemical mechanism that we've already talked about. So there's the hydride transfer. This is in the oxalate as substrate. You have to have a hydride transfer from NADH and a proton transfer, proton donation to that hydroxyl group. Okay. In the reverse direction, It's interesting, but the histidine, this histidine, has to be unprotonated for catalysis because there's an OH here, and the role of this nitrogen is to abstract the proton from the OH. And the hydride transfer, of course, goes in the opposite direction. And we can talk about some things related to that later, but that has implications for what the PK of this histidine is and what the role of the aspartate is. The aspartate changes the PK of the histidine, obviously. And there are probably some other factors around this region of the protein that are involved in substrate, i.e. cofactor and carboxylic acid substrate binding that relate to the PK of this histidine so that it's primed for the right steps in the reaction. So at the bottom here is a web logo plot of the conservation in the flexible loop region. And obviously it's got 2 of the 3 arginines. The third arginine is way down in the sequence. And here is that asparagine. But there are some other residues that are definitely highly favored in certain positions. So there's a proline here that's highly favored. This aspartate, this leucine, this methionine, this glycine. If you're not used to a web logo plot, the size of the letter shows the propensity for that residue. So here, for instance, proline is favored over glutamate over I think that's an aspartate down there. But this shows the mobile loop region in terms of where it sits in terms of, so the red on the blue subunit and the sort of salmony pink on the opposite subunit is the mobile loop. And we're going to spend a few minutes now talking about the movement of this mobile loop region. A lot of enzymes have loops that close over the active site, partly to exclude water or increase the hydrophobicity. And that's certainly some of what's going on in here. And we've actually looked at crystallographic orders and can track the water movement in the active site as the loop closes as well. So this is a very complicated picture, and I'm going to sort of step you through it. So again, in red is the mobile loop of the unliganded subunit. So this is the unliganded or open form of the loop, and here it is in the salmony pink on the other subunit. When cofactor binds, the loop closes from the red position to this sort of raspberry colored position on the site subunit that the cofactor, so the cofactor is bound in here. The loop closes, not completely, but closes somewhat. On the unliganded subunit, ignore this for a minute, I'll explain what that is in a moment. On the unliganded subunit, it doesn't close, it doesn't really change position very much. So we go from fully open to partially closed in the raspberry. The green is what happens when the malate binds in. It closes a little bit further and repositions itself on this subunit. Again, it doesn't do much on this subunit. Okay, so you're sitting there looking at this and you're thinking to yourself, well, what on earth is this yellow loop up here? And that's interesting because what seems to happen is this is loop movement on this subunit where we put the ligands only on this subunit, when in addition, we let cofactor bind to the opposite subunit, okay, then this is the equivalent of the red to salmon, is the sort of pink to yellow. So there's loop movement on this subunit now because we put in cofactor on this. But interestingly enough, what happens is the loop goes from the green position to the yellow position on the opposite subunit. So cofactor binding to this subunit has caused a change in the loop position on the subunit that's had catalytic reaction taking place, or may have had catalytic reaction, because it's got all the components. And if you think about it, this opening of the loop may be part of a process of letting the products release from the subunit where catalysis. No catalysis has occurred on this subunit because we haven't put malate into this subunit, or oxaloacetate rather, depending on which direction it's going. So the loop movement is rather complicated. So the overall reaction catalyzed, and a lot of people forget this guy, but there's a proton involved in this. It's the proton that's abstracted onto the histidine or donated from the histidine, but there's a proton in the overall reaction. So malate NAD goes to oxaloacetate NADH and a proton. The equilibrium constant of the reaction under physiological conditions, I'm going to emphasize that those are defined as 38 degrees centigrade, pH 7, and constant ionic strength, favors malate and NAD. Okay? And of course, if you change the pH, you're going to change the equilibrium position because A, there's a proton in the reaction. So of course, if you change the concentration of this, the Chateliers is going to change the equilibrium position. And then this just summarizes the catalytic mechanism again. And what I really want to get to is this diagram, which shows you those critical positions in crystal structures from a variety of different sources. And so the histidine, no matter what the isoform, is always in the same position relative to the malate and the cofactor. R196, which seems to be a Mainer coordinator of the malate or oxaloacetate, is always in the same position, and D193 is always in the same position. So, that geometry of the active site configuration holds for pretty much any malate dehydrogenase that exists. In terms of the catalytic mechanism, we've had a long-standing interest in understanding the mechanism at a quantum mechanical level. And this shows some what I call QMMM studies. And we're going to talk about molecular dynamics later on. But QMMM lets you look at bond breaking events, whereas molecular dynamics just looks at movement of things. So this gives sort of an overview of, don't worry about the details, but what we do is we set up, based essentially upon the crystal structure positioning that I just showed you, because it's consistent with all MDHs, we set up a box that we're going to do our QMMM on, and then we start doing QMMM in this so-called truncated model. And what it does is it lets us look at bond-breaking events, the hydride transfer, the proton donation, etc., and work out from the QMMM calculations what are the rate-limiting steps. And this just gives some typical data. So, I mean, unless you're into QMMM, don't worry about it, but it's possible. And the importance of being able to do it in a region like this by QMMM is that you can now add other residues into this to determine whether or not they impact the transition state and the reaction coordinates, things like that. So I'm not going to go into detail, but in the PowerPoint, if you want to read, it's got some of the detail there. What I want to do now is switch to the kinetic mechanism. And, enzyme kinetics are easy to do because in the reaction, the NADH absorbs light, whereas NAD doesn't. So you can follow the kinetics very, very simply in either direction with either absorbance or fluorescence. Fluorescence is probably better in the NAD to NADH direction. Absorbance is better in the NADH to NAD direction. But, it's easy to do kinetics. And so I want to spend just a moment or two talking about kinetic mechanism. There are a variety of potential kinetic mechanisms. So this is what's called a compulsory order steady state mechanism, where the compulsory order is first substrate comes on, forms a complex, second substrate comes on, forms a ternary complex. Chemistry happens, you get a product complex, and then the products are released in order. So that would be a compulsory order steady state. You can also have what's known as a compulsory order rapid equilibrium, where these steps are in equilibrium with one another. You'll notice here, these are unidirectional arrows indicating steady state can be established. But these are reversible arrows indicating that E and ES1 are in equilibrium with one another, and ES1 and ES1S2 are in equilibrium with one another. And then you have the catalytic step to give you products. So that's what's called a rapid equilibrium mechanism. But again, it's a compulsory order. Now you can have a steady state or a compulsory order that is random, so-called random, and many dehydrogenases show a random order where it doesn't matter which substrate binds first, then the second one binds, and the same with product release. And again, it can be compulsory order, it can be random order. Difference between random order is these steps are in equilibrium and can be represented by equilibrium constants, whereas these steps are in steady state are represented by rate constants. And then the other type of two substrate mechanism you can have, and sometimes this is referred to as a ping pong mechanism, where one substrate binds, one product, you get some chemistry occurring, one product is released, you get an enzyme substituted, enzyme, a second substrate comes in. So this is typical of a group transfer type reaction. Malate dehydrogenase is not an enzyme substituted mechanism. It could be a steady state compulsory order, or it could be a random order steady state or equilibrium versions. So there are four potential kinetic mechanisms. And again, different isoforms or different mutations could change the kinetic mechanism, which is why I'm talking about this at this point. So if you want to do kinetics, there's a standard way of approaching dissecting whether or not you've got a random or compulsory order or a steady state or a rapid equilibrium type mechanism. And that involves this thing called the generalized rate equation, which is this equation here. So this is the enzyme concentration, the measured initial rate. And then there are four what are called initial rate parameters, phi0, phi1, phi2, and phi12. And phi1 has got this substrate A in it. Phi2 has substrate B, and phi12 has substrate A and B. These parameters can be experimentally determined without making any assumption about what the mechanism is. And that's what distinguishes this type of kinetics from most other kinetics. Most other kinetics, you assume a mechanism and fit your data. Using this approach, you fit the data and then look for consistencies with certain mechanisms. So this is a much more rigorous way of approaching enzyme kinetics. And I've just shown you here, all of this is described in a book I wrote many years ago and is still floating around, but now the whole of the book is available on my website, and this is the link to it. But essentially, you determine these phi parameters by doing a series of Michaelis-Menten kinetics, where you'd convert them into Lineweaver-Burke plots, so that E over V is the equivalent of the 1 over V against 1 over substrate concentration. And you get a series of lines. If this is substrate A varied, you do it at a fixed concentration of B and you get a line. But then you change the concentration of B and repeat it. And you get a series of lines, each of which has got a slope and an intercept. And then you plot the slope or intercept against the reciprocal of this substrate, the B substrate. And then the slopes and the intercepts of these give you values, numerical values for the phi parameters. And of course, because you've done data fitting, you have numerical values, plus and minus standard deviations, etc., etc. And those are diagnostic of the mechanism as described in this chapter. The important point I want to make, since some people sort of stop at just doing regular Michaelis-Menten kinetics and calculating a Km and a Vmax, the important point is that Km and Vmax are dependent upon the concentrations of both substrates, not just the one you vary. And so this gives you a way of getting a true Km for each substrate and a true Vmax, because this is the Vmax parameter. And then the true Kms are ratios of these two or these two parameters, etc. And so you can get the true constants, which when you're comparing isoforms or mutants is really important, because if there's a different dependency caused by a mutation or caused by a different isoform, you're not calculating real kinetic constants. Okay, so this again is the summary of the ordered ternary complex mechanism that most malate dehydrogenase wild types have been shown to have. Okay, NADH binds first, oxaloacetate, proton comes in, catalysis occurs, proton comes off, malate comes off, NAD comes off. Now, one interesting thing in the literature is it's been suggested that malate dehydrogenases show what is called a reciprocating subunit mechanism. So as we've talked about, all malate dehydrogenases have at least a dimeric structure. Tetramers are a dimer of dimers, remember. And a reciprocating subunit mechanisms, there's two types of reciprocating subunit mechanisms that have been shown to apply to other dehydrogenases, to things like ATP synthase, etc. But so this just is a schematic. You form enzyme substrate complex on one subunit. you get reaction to an enzyme product complex. Substrates bind to the empty subunit in this, and then reciprocating subunit triggers product release. So now this one has a conformational change triggered by substrates binding to the previously empty one. Products get released, and you're back to this starting complex and just keep going around the cycle. That's one type. That's where the reciprocation triggers product release. The other type is where, again, you get enzyme substrate complex on one subunit, but now it forms on the second subunit in the dimer, and the reciprocating subunit triggers catalysis. So substrate binding to this one triggers this one to become catalytically active and form a product complex. And now products leave, and of course you're back to our original situation. Substrates bind here, triggers product formation on this one. So the two different types of reciprocating subunit mechanisms, reciprocating subunits trigger product release, or they trigger product formation. And nobody knows which is really the case in malate dehydrogenase. But that brings back what I was saying about the loop movement. This is just the same slide again, same couple of slides, just to refresh your memory that I showed you that cofactor binding to one triggers a conformational change on the empty, on the subunit that's already got something bound to it. So, there's evidence for such conformational changes. So that these are in support of proposed reciprocating subunit mechanisms. And this is just a little schematic we published a few years ago that shows how that might be involved in a variety of phenomena that are known to occur, because conformational changes can be fast, they can be slow, they can be whatever. And this summarizes the reciprocating subunit mechanism, what we think might happen when citrate regulation is involved, and what we think might happen when substrate inhibition. And when you vary oxaloacetate concentrations, you get what's known as substrate inhibition in most isoforms, and that we think might be part of a reciprocating subunit mechanism. So lots of cool things you can do with both kinetics and conformational changes and things like that. Interestingly, citrate, and so this is back to the loop when citrate is on here, so the open loop. When citrate, it closes even further. And interestingly, citrate, when it's bound, that's shown over here. So the loop in citrate closes even further than it does in the normal catalytic cycle. But when citrate is bound, if you try and model NAD or NADH in, you see that part of citrate really competes directly with the cofactor. Down here, it's competing with the dicarboxylic acid substrate, because of course citrate is the dicarboxylic acid substrate with an extra pair of carboxyl group carbons on it. But kinetic studies show that citrate is competitive with both the dicarboxylic acid substrate and with cofactor, and that's consistent with the structures. So because of the involvement of a proton and pH-dependent studies, I just wanted to point out that some interesting things you can do with pH-dependent studies, and I'll show you some kinetic data in a moment. But you can make differentially protonated structures of the protein using H++, which is a computational program that allows you to look at the PKs of protonatable groups on a protein and what impacts them. But it also lets you create differentially protonated structures. And so here I've just shown you quickly how you do that for those of you that might be interested in this sort of thing. So when you open up H++, you put in your structure, you start processing your structure. First thing that happens is a box that opens that lets you customize things. You can put in salt concentration, you can put in dielectric constants for the solvent for the interior of the protein. But more importantly, you can put in the pH that you want it to do the calculations at. So here I put in pH 8, but I could just as easily have put in pH 5 or pH 6 or pH 7 or pH 8 or pH 9 and let it do it gives you does all the calculations. And the interesting thing is, in terms of the usual output, and these two are the things that list all the PKs and the residues that impact the PKs. If you've never used H++, it's a great in-class exercise as well as research tool. But the interesting thing is it gives you a PDB file that is protonated according to whatever pH you told it. So if you ran a whole bunch of different pHs in H++ in this box, you would get a whole bunch of structures that are protonated according to the pH you told it to use. And the interesting thing about that is that it lets you save those protonated PDB files. And now if you want to use them in Docket or surface properties or whatever, you can do a pH dependence experiment in silico. Or if you want to do QMMM, you can do, you've got the ability to do pH dependent calculations of binding, of conformation, of stability, of anything else you want to that you can do from the PDB file. So it's sort of a nice add-on to what you can do. But if you just do pH dependent kinetics, this is published data, and you see that depending upon what you use as substrate, you get, with male H, you get the blue curve. And, if you ever look at pH curve, there are a couple of PKs in there. If you use oxaloacetate, you get the red curve. And again, there are a series of PKs that you can calculate from the curves. And you can get, the PKs of the histidines, and, but it helps you work out whether or not the PK is going to be protonated. The histidines can be protonated, unprotonated, etc. And so this is going to lead us into the whole allosteric regulation thing about citrate being a potential allosteric regulator. The conundrum is that nobody has ever described a citrate allosteric site or an allosteric site in malate dehydrogenase for any ligand. The only sites that exist in crystal structures are the so-called orthosteric or active site binding regions. Okay, no allosteric sites have ever been found. Okay, so this reciprocating subunit model is part of an explanation of that conundrum, where one subunit in the dimer, in essence, acts as an allosteric regulator of the other subunits. And I'll show you some stuff that's relevant to that in a few moments. this is just a refresher again, because as part of the studies, we were looking at, the roles of the arginines. And we noticed in binding studies, in computational binding studies, and we noticed that consistently there seemed to be another arginine that may provide an additional binding site. And so this is a binding study where this is binding at the active site, which is determined by the canonical 3 arginines. But there's another binding site out of the active site down here that seems to center in on arginine 249. And so while we've done computational mutations of that, we've made the experimental one as well now, but we haven't finished doing the kinetics and binding studies with that. But it's conceivable that this down here is a sort of a waiting area for the dicarboxylic acid substrate. And it has to sort of jump from here into here. That's all speculation, but it's fun to think about, especially when, and we'll talk about metabolons and things like that later. Okay, so I think I got to this one when I did the quick overview before, but this is where we switch into thinking about the dimer interface and its role and its structure and some of the fun things you can do with that. So this just shows you the different regions of the dimer interface. And so there are a bunch of regions and they crisscross across the interface. So this shows you which regions talk to which regions. So there are 5 regions, and the region 2 talks on the other subunits to region 2 and to region 5. Region 3 talks to region 5 and region 4, etc., etc. So those of you that like to think off the wall and have fun thinking about weird and wonderful experiments, depending upon what sort of equipment you might have available to you, I wanted to just at this point show you a cool approach that we're developing to look at the strength of subunit interactions in a protein such as malate dehydrogenase. And this uses atomic force microscopy. So those of you that aren't familiar with atomic force microscopy, there are 2 modes of atomic force microscopy. There are so-called tapping modes that are used to determine physical size, but there are pulling modes that are used to determine the energy of a structure. And we've adapted the pulling mode of atomic force microscopy to look at potential subunit interactions and the strength of the stability of the structure. So what you do is you use relatively low pH, because it's known that malate dehydrogenase goes from dimer to monomer at relatively low pH. You use a gold surface because in the monomer there's an exposed SH group and that helps it bind. To this you then come in with the pulling probe in AFM that's got a subunit on it, and it can form a dimer, and then you start pulling. Or if you don't coat the tip with the monomer, you can just look for the one you've already laid on the surface and form a connection and start pulling. So you can look at stability of the protein itself, or you can look at stability of the interface. And as you pull, you see changes in the force that equate to either unfolding or monomerization, etc., etc. And so it just gives you another cool way, especially if you're teaching a cure in a biophysics course, then AFM, if you've got one around, is a cool way to think about doing studies, and you can combine it with mutations and things like that. So I keep coming back to this summary slide just to refresh your memory of the overall structure. Here are some references to some of the QMMM type things we've been doing, and Hugo was pioneered that and did a really nice job with that and is still working with that as well. So this is a summary of the kinetic parameters, those 5 parameters. And there's a relationship of the 5 parameters to the rate constants that were in the various mechanisms, and the relationship to the true Vmax or Kms for the 2 substrates, and depending upon the mechanism. But as you can see, they're diagnostic for a particular mechanism. The interesting thing about kinetics is when it comes to thinking about rate limiting steps, they can either be bond breaking or they can be conformational. And either which way, if you use isotopically labeled substrates, so if you put the isotope in the bond that you think is bond breaking, rate limiting, then the isotope will have an impact on the rate, etc. Whereas if it's conformational, is the rate limiting step an associated conformational change, you won't see an isotope effect. And again, we've done some cool isotope effect experiments combined with stock flow kinetics as well as regular Michaelis-Menten kinetics. And it's an interesting way to think about if you made a mutation that changes a rate limiting step, did it change a conformation associated with that, et cetera, et cetera. And this just reminds me that, and again, I always talk about this totals approach, but, enzyme kinetics is a great, but there are many other ways where you try something, observe what happens, think about what happened, adjust and try again, and then leave lab smiling for totals. And the text just explains that. You've heard me talk about that before. So let's think some more about allostery now. So in terms of allostery, we've already talked about this diagram. And this is just to refresh your memory that there are many points of contact across the dimer interface. You can also do this sort of analysis in the tetramers, and we have done, as you'll excuse me, see a little bit. We've done this for the tetrameric forms as well. And this is a typical Hawk-Dock analysis of the dimer interface, in this case, of human mitochondrial MDH. And just to refresh your memory, if you've not done Hawk-Dock literally steps across the structure of the protein and says, is a residue making an interaction with something on the other subunit? And then it quantitates it either as repulsive with a positive delta G or attractive with a negative delta G. And so you can see these are the various 1, 2, 3, 4, 5. We break this one into 2 regions. So those are the 4 regions. In human mitochondrial, K27 forms a repulsive interaction, but all the rest are attractive interactions. And here is a representation of those many interactions. This is from a program called HINT, which is a commercially available program. And we've used HINT with a number of different structures. If you're ever interested in getting hold of HINT data, but don't have the many thousands of dollars it takes to actually buy the program, I can provide you with HINT data for representative isoforms. But here, blue is attractive, red is repulsive, and it shows you all along this interface, even where the major residues are, there's lots and lots and lots of little interactions going along. And that can be quite interesting when you look at different isoforms. So I showed you human mitochondria that had a little repulsive one, whereas Plasmodium falciparum has E18. Um, this shows you where E18 is at the interface, and you can see you got this glutamate that's juxtaposed with the same glutamate on the other subunit. Two negative charges, obviously repulsive, big repulsion. But you've got a whole bunch of attractive interactions, and then this one big. You got a couple of little ones. This, and this shows that human mito and human cyto really don't have anything very large down here, but have very similar overall structures of the interface across the rest of the interface. And we've made some cool mutations around this region to try and change the electrostatics. So what we were interested in doing was probing the role of the electrostatics here on activity and subunit interactions. So we made a whole bunch of mutations around the E18. We left E18 there, but we made mutations that would change the electrostatic interaction there. And we looked at those. And this shows you, again, a hawk dot type thing. It shows you that there are some residues around here that might impact the local environment around E18. So we made mutants of these 4 positions, and then we looked at the quaternary structure through size exclusion chromatography. And you can see the wild type is the tetramer structure. When we change L22, we shift this a little bit, but not very much. L22N has, I think you'd agree, a relatively normal quaternary structure. L19N, and these were designed to increase the polarity. L19 totally wrecks the quaternary structure, and this is a monomer form. Whereas I15Q, again, changing the polarity, gives us a dimer form. And Q11, when we increase the non-polarity, we make it less polar, also disrupts. And these two look fairly similar. So pretty much things around that E18, that repulsive E18 interaction, have dramatic impacts on the quaternary structure. And Danny Oenders, who did all this work, is just in the process of finishing up all the kinetics and stability studies on all these mutations. And we're hoping to have the paper submitted in the next weeks or months at most. And Danny's done a great job on this. So that brings up the issue of, are there molecular switches at the interface that trigger subunit interactions? And we've done a bunch of studies on that. These were started by this group of students, and especially Michael Schwabe, who just got his PhD from Northeastern and is now a postdoc at Harvard, and Nina Maria Garcia. They did a fabulous job making mutations and looking at the interface. And they noticed that there were a bunch of different sorts of cross-interface interactions. So in particular, this D18, D87 forms an interaction with the positive pole of the helix dipole across the interface. You often think of just side chains being involved in interactions, but secondary structure, especially helices, have dipoles, and so dipole charge interactions can be very important. And there's an important, or we think important one, D92 and Y272 across form and interaction across the interface here. And so we've done a bunch of studies making mutations around the interface here, and all of the organelle forms have these conserved S266 and L269. We are fairly convinced, and again, a paper is being written up on this at the moment, we're fairly convinced that S266 forms an interaction back into the active site, whereas L269 forms the switch across the interface. Okay, so we mutated both of those. So for instance, this shows S266A mutation, L269A mutation with NADH as so you get normal Michaelis-Menten kinetics. When you make S266A, you get a nice Michaelis-Menten curve. The parameters are a little bit different, but not much. There's not much of an impact of that on Km for NADH or S266A. But L269A, totally different Michaelis-Menten plot. And in fact, you can't even calculate a Km for NADH from this. It turns it into a linear plot. And so L269A has a dramatic impact on saturation by NADH. When you look at the same mutations with oxaloacetate, interestingly enough, you find the exact opposite. Okay, S266A dramatically changes the saturation by oxaloacetate, but L269A has a very similar. The KM's changed a bit, but not very dramatically. The dramatic change is the S266A. And so those residues form, we think, this molecular switch that transmits information. To follow up with that on a tetrameric, we did some studies with Icelandicus ignococcus, or Ignococcus icelandicus, I should say, malate dehydrogenase. This is a tetramer, but it's got a very different turnover number, low specific activity, and it shows strong substrate inhibition by with respect to oxaloacetate, whereas the watermelon somal enzyme doesn't show terribly strong substrate. It shows some substrate inhibition, but nowhere near as dramatic as with the Ignococcus enzyme. And so we constructed a series of things where we switched small regions that we'd identified as potential interactions across the interface. We switched them. We made these what are called knock-in mutants, where we knocked in regions. For instance, we knocked in that serine and leucine. This is 266 and 269 in watermelon. They're absent in the Ignococcus, so we put them into the Ignococcus. And then, we put in D92, and we put in this, and then we put in Y273, and we looked to see what happened. And on the somewhere. This is the wild type specific activity. This is the SATL, dramatically lower. These guys actually increased. So now we're seeing a difference impact of different parts of the interface. This we anticipated, but the interesting thing about this was that, yes, it lowered the activity, but look at the impact. This is wild type Ignococcus showing that strong substrate inhibition. You see it out even as low as 0.5 millimolar, whereas usually you have to get up to about 10 millimolar before the wild type watermelon, for instance, starts to come down. Whereas when you put in that watermelon component of the switch at the interface, you take away that low concentration substrate inhibition. So that led us on another direction, and that was we tried to trace the information flow between the subunits using molecular dynamics. And we started by doing things that we knew were different and were inhibited. So we did molecular dynamics simulations in an AB, this is watermelon, dimer, with nothing bound, and compared it with citrate binding. And we looked to see what happened. And this summarizes a whole bunch of data. So these are the interesting residues that showed up on subunit A. These are where they're located. Mutated with impact, i.e. have we been able to mutate them? May have an impact on the activity, yes. Residues with highest change in dynamic correlation, i.e. these are the things that move the most. Where were they, and have they been impacted with impact? That's on subunit A. On subunit B, we found a whole bunch of things, and these are just summarized in the same sort of way here. And the next diagram shows what we think is the flow of information between the subunits. So the things shown in spheres on the two subunits, this is the subunit we put the citrate on. These are the things that moved, and you can see this sort of swoop towards the interface region. And then across the interface, these are the things that moved on the other subunit. There's sort of to the reciprocal region. And so, we've traced the flow of information induced by citrate. And we're in the process of finishing up some molecular dynamics work in conjunction with Chris Bernston at JMU, looking in more detail about the various different complexes in the tetrameric forms to look at the tetramer interfaces as well as the dimer interfaces. But there's a ton of interesting stuff you can do, and this can lead to creating hypotheses about the role of particular residues and what they might be doing in designing mutants, etc., etc. So there are a whole bunch of inhibitors, potential inhibitors, or activators. And I divide them up into two categories: what are orthosteric ligands, and these are things that bind at known binding sites, versus allosteric ligands, and we've already talked about citrate. Succinate seems to activate and maybe activating through an impact on the reciprocating subunit mechanism, because we have no known allosteric site for these, even though they show allosteric type behavior. And so I think one has to be careful distinguishing between allosteric type behavior and allosteric sites. Although these show allosteric behavior, there's no known sites for them at this point. And here's some references about those that we published over the years. As you can tell from The earliest of these dates back a good 15 years now. We've been looking at these sorts of things for a long time. And so what I want to end up with is this idea of cryptostery as opposed to allostery. And I'm going to define what I call cryptostery in just a moment. And cryptosteric sites most definitely exist on malate dehydrogenases. The potential for what I call cryptosteric drug design. I just gave a presentation at the Protein Society last week, and I'll put up my PowerPoint from that as well. But to cut a long story short, a cryptosteric site is a site that exists on one isoform, but not another isoform. And it could be because there's a loop. So down here, this is a pairwise sequence alignment of two forms. You know, here's a loop that's in one form that's missing in another form. And so that's a potential cryptosteric site. But it could also be, so a region here, where there's a significant difference between the amino acids that are on one form and the other form. Those are potential, what I call cryptosteric sites. And if you followed our presentations and publications, you know that we've shown this in the Plasmodium falciparum, malate dehydrogenase. Some very nice work started by Natalie Botros, who actually won the ASBMB undergraduate poster competition with this work. But she did the sequence alignments, identified potential cryptosteric sites, and this shows the potential cryptosteric site, the active site region. She made mutants of this and showed that, yes, there was some functionality. She did sequence conservation, again, web logo type things to show that the cryptosteric site was conserved across all the pathogenic species of Plasmodium falciparum. She mapped binding pockets with POCASA, which lets you get, in essence, the dimensions, the depth and dimensions of the site. She looked at the electrostatic surface properties of that. And here's the cryptosteric site. There's the pocket. You can see there's quite a deep pocket there that she mapped nicely. And then this is showing the actual pockets as detected by POCASA. And she went on then and did a really nice job with looking at what might bind in this pocket using computational take on multiple solvent crystal structure. This was developed by Carla Matos and Dagmar Ringer, and we developed a computational approach using SwissDoc that does the same thing. It tells you down here is a summary of what binds, what fragments bind into various subparts of the cryptic pocket. Then you can see there's a whole bunch of things bind. And of course, the mission now is to combine fragments and find an effective ligand for this pocket that disrupts the activity of the protein. So we showed, this is just a summary of some of this in visual, and the reason for that is to show you that the cytosolic has very different, as does the mitochondrial. And so it's a way of getting a unique interaction with the cryptic pocket. So that's the basis for cryptosteric drug design. And we've done that with P. falciparum. We've also got ongoing projects with P. gingivalis and C. albicans which are part of the oral microbiome pathogens. We have a lot of pre-dental students, and Millie and Haley, you were both interested in dental school and dentistry and wanted to do something that had something to do with the oral microbiome. And so this is a way of potentially designing inhibitors against oral microbiome pathogens. And so what I want to finish on is talking a little bit about metabolons. I referred to metabolons earlier, and the reason for this is targeting metabolons is potentially a productive way of designing drugs to disrupt activity. So this shows you malate dehydrogenase. You now all instantly recognize malate dehydrogenase. But it shows the metabolon. This is the mitochondrial metabolon that's formed with citrate synthase, with fumarase, and with aspartate aminotransferase. So a lot of people just look at this one, but in actual fact, it's really a complicated metabolon that's been looked at. There's some nice publications out there about the evidence for the metabolon. But the thing that caught our eye was that some of the mutations in malate dehydrogenase, human malate dehydrogenase mutations that are associated with early onset severe encephaly, line themselves up at regions that make them look interesting for metabolon formation and maybe disrupting metabolon formation. And so it was appealing to think about that and to look at where potential druggable pockets, and this just shows that same metabolon with potential druggable pockets identified by Bocassa. And you can see that they line up in many of the same regions that those mutations were. So there's the potential for future drug design. And this just shows the potential cytosolic metabolon between malate dehydrogenase and again, some sites of post-translational modification, O-gluconylation, which has been shown to have an impact on activity, or lysine acetylation, which has been shown to have an impact on activity, are again at interesting places on the metabolon. So there's the potential of developing drugs or developing things that block such interactions. So I'm going to finish up with just some summary slides that emphasize the interface between computational approaches and wet lab approaches, since they can be used synergistically to great effect. And I want to start by just reiterating one of the reasons why we all are trying to do cures is because vision and change, promoted the attributes of what they call a scientifically literate student, apply quantitative reasoning, collaborate across disciplines, understand the relationship between science and society, apply the ability to apply the process of science, use modeling and simulation, and make use of the interdisciplinary nature of science. And you've probably all seen this slide before, or a spin on this slide before, that has the various different elements of research that you should incorporate into your cure: relevant scientific background, hypothesis, presenting a proposal, designing and execute experiments, and teamwork and collaboration, reproducibility, data analysis, and project presentation and peer review. And this just shows how you can use virtual components, virtual approaches to look at all of these things. And this then matches computation approaches to protein structure function and wet lab collaborations. So on the left-hand side of this, here are the protein structure function relationships related primary, secondary, tertiary, quaternary, protein stability, you can read through them all, etc., etc. So these are all the sorts of issues, scientific and otherwise, that you can think about in a project related to protein structure function. I've aligned here the various computational approaches, and you'll see there's lots and lots of computational approaches for each of these, all of which are on the website, and there are videos about using them. And I think Chris has just done a video about molecular dynamics. It's a nice adjunct to the website as well. But yeah, we previously had done ones on all of these other things. And then these are the wet lab collaborations that you compare them with. So for instance, you know, if you've got access to mass spectrometry or a collaborator with access to any of these, you compare them with primary, secondary structure, you know, again, it's a whole bunch of wet lab techniques, you know, pH dependence, isotope effects we talked about a little bit. But there's all sorts of other things. I showed you data for size exclusion, but chemical cross-linking to look at protein-protein interactions is a very powerful technique. And again, we published on some of the protocols on those. But, you know, all sorts of different wet lab experiments that you can compare them with. And so this is just a brief summary of some of the sorts of cure projects that you could do either completely virtually using computational approaches or pair them up with wet lab approaches, as we just talked about. So, you know, drug design, looking at the role of what are called second sphere residues in specificity, in catalysis, etc. Looking at the evolution of allostery. We've just started a project now where we look at cryptosteric sites as forerunners of potential allosteric sites, things like that. So this is, again, a second sphere idea, things that aren't in the orthosteric site but may be influencing the orthosteric site. And, you know, subunit interactions, and then the evolution and the role of quaternary structure, etc., etc. And then these are some of the advantages of adapting either completely virtual or virtual components into cures. And I've broken this up into two sets, the faculty perspective. Good thing about virtual computational approaches is there's no setup activities. You just need a computer. There's no expensive equipment needs. You just need a computer. There's no expensive reagent needs. You just need a computer. They can be taught synchronously or asynchronously. And so they lend themselves to out of regular classwork time as well as regular classwork time. And they can all be related to either collaboration with wet lab or to ongoing research activities. And they're often a simple way to test out potential new research activities. Because they're a cure, they have all the same advantages for the student with, you know, all the things that we preach to our students they should be getting out of lab courses. But because they can be done off campus or asynchronously, they can be, there's no need to travel to campus, there's no added lab fees, etc., etc. So there's many ways that they are of value to an increasingly diverse population of students. And I'm going to finish on just one quick, this was from the Protein Society Educators Workshop that Jessica and I and Dan Rees and Josh Beckham did last week. And again, I'll put the whole of this PowerPoint, but the thing I want to relate quickly is the key to a cure. So here we are, the key to a cure. And I should have said keys. So the first key is if you or anyone else knows the answer, it's not research. And if it's not research, then it's not a cure because the R stands for research. So, this is what distinguishes cures from guided inquiry and those sorts of things. You've all probably found things like this on the website. We've developed rubrics for all the different elements of research. So that's just a list of them. And again, this is all on the website. How to go about developing ideas for a cure and developing a syllabus. And again, resources are up on our website. And the totals approach, the rules of 3 approaches to hypothesis development, things like that I've talked about before, so I won't waste your time going over it again today. That's just there. This is the one I wanted to end up with, that cures are a high impact educational practice. And this is my favorite quote: many high impact educational practices tend to involve learning at the edge of chaos. This is the originator of that quote. And our job as faculty teaching a cure is not to try and get rid of the chaos. It's to ensure that the students learn at the edge of chaos, because the more chaotic your lab is, the more the students are going to learn from it. So resist the temptation to tell them what the hypothesis is or give them a project. Encourage chaos and let them develop their own ideas and do their own literature searches and things like that. Don't just hand them a review and say, hey, here's the background. They learn by doing it themselves. And so learning at the edge of chaos is the key to a cure. And with that, we are finished. And so we've got one person that's been patient and sitting through. And you got any questions? If not, I'm going to stop the recording anyway.